Welcome to episode 3 of my molecular biochemistry series. And this one's going to be a bit longer, but we are focusing on nucleotide synthesis. So previous videos, we looked at the structure of DNA in detail. We looked at the molecules that make up the DNA helix. And now we want to understand how you get these nucleotides there, because they don't just appear from nothing. Our bodies, our cells need to synthesize them using various different chemical reactions or a metabolic pathway. We also looked at the DNA in context. We saw that it doesn't just sit in isolation, but in fact coils around a protein called the histone. And it does this over and over and over and over and over again until you get a supercoiled structure that we commonly call the chromosome. We also saw that there are some epigenetic factors, which just means that we are modifying the transcription of the DNA without changing the nucleotides or the structure itself of the DNA. We are just either coiling it so that transcription doesn't take place, or we are uncoiling it so transcription is more likely to take place. And we saw that by either removing acetyl groups or adding methyl groups. But now we want to spend some time thinking about how we make these nucleotides. And there are two major pathways to do this. We have de novo synthesis, which branches into two more metabolic pathways, or we could use a salvage pathway. And we'll look at the salvage pathway first, but I want you to know that we rely on a very important molecule called PRPP, and we get it from starting with the glycolysis pathway, although nothing ever really starts anywhere in metabolism. But you get glucose from your blood, enters glycolysis, you get G6P, but this enters what we call the pentose phosphate pathway, or PPP, which is known for biosynthesis when you're first learning it. And this results in ribose 5-phosphate, and then that gives us PRPP, which stands for phosphoribosyl pyrophosphate. And as you can see here, it plays a role in both pathways and is pivotal to nucleotide synthesis. So in the salvage pathway, we see that it enters here, it enters right here. And keep in mind that these are very simplified diagrams. There are many reactions involved in all of these pathways that are not written here, but I am focused on the main portions of each pathway. So in the salvage pathway, once nucleotides fall off or part of activities and are left over, we can salvage them, and their bases become available to create new nucleotides. And these free bases could also become nucleosides, and it enters the pathway. Specifically, at some point it will reach HGPRT, which is an enzyme that is critical to making nucleotides. From a medical perspective, you can look at various different diseases or syndromes or disorders that can result from lack of nucleotide synthesis. One of them is adenosine deaminase deficiency that results in SCID, which is just a severe combined immunodeficiency. And some of the symptoms that result from this are an impaired immune system. So some infants may suffer from this, and they'll have to be put in a sort of bubble because their immune system cannot fight off any germs. They can't fight off bacteria or viruses effectively, and they are at risk of becoming very, very ill and even fatal. Additionally, another syndrome is Lesch-Nyhan syndrome, which is the absence pretty much of this very important enzyme in HGPRT, and its symptoms can be remembered by using HGRT, such that you could have hyperuricemia, you could have gout, You could be pissed off all the time. You're very aggressive. And then you could have red or orange crystals in your urine. Or you could have tense muscles, also known as dystonia. So those are some of the symptoms if you have Lesch-Nyhan syndrome. Of course, we could flip things over, and if we look at de novo synthesis, instead of being impaired, instead of having a disease or disorder or syndrome resulting from the lack of nucleotide synthesis, we can manipulate this in a way that will actually treat various illnesses. For example, you could use methotrexate to treat cancer, because by impairing or inhibiting nucleotide synthesis, we can prevent cell proliferation, specifically cancer cells or bad cells. Alternatively, you could have ribavirin, which will be an antiviral and prevent the nucleotide synthesis for viral replication, which is useful if you have a viral infection. And it does this through guanine, inhibiting guanine synthesis. Of course, you could also have leflunomide, which is relevant for arthritis. So if you have an overactive lymphocytes, we actually don't want as many lymphocytes. We don't want the level of inflammation you have in a certain region. You could think rheumatoid or psoriatic arthritis. And so in this pathway, you can have two branching. You could have one where you get to build the ring first, and eventually you reach carbamyl phosphate synthetase 2, which synthesizes carbamyl phosphate. And of course, PRPP enters, and we get our pyrimidines, which, if you saw in my first video, I broke down what those are if you're unsure at the moment. In addition, you could go through this branching and build purine synthesis, and it also uses PRPP right away, and you get folate, ATP, and GAG, which stands for glycine, aspartate, and glutamine, to create IMP, which then goes through various different other reactions, and we finally get our purine. So these are the de novo synthesis, which requires a lot of energy compared to the salvage pathway. And this is a great overview of how we get our nucleotides, which drives DNA transcription. And with that, I'll end there, and I'll see you guys in the next one.